and it is my privilege to introduce Angelica Lang for the seminar today. Angelica is a fifth-year graduate student in my lab. She bravely joined the lab in 2021, right after we moved from the NIH. She was the first grad student who joined my lab, which you can ask her if she thinks that turned out okay. I think it was all right. But she's done beautiful work. So Angelica did, to back up, she did her undergraduate work at the University of Kansas. And while she was there, she worked with Eric Lundquist, where she learned to use C. elegans to study a really beautiful phenomenon, neuroblast migration. And she has a first author paper from her undergraduate work, two author, first author paper from an undergrad, which is impressive. And then she came here for graduate school in the genetics program. And I was really lucky and really excited when Angelica decided to rotate with my lab, and honestly, a little bit shocked. I was really new, and I was really excited to have a rotation student. And then immediately upon starting her rotation, it was very clear Angelica was going to be a star and she was going to excel. And so when she decided to join my lab for her PhD, I was very excited. And she jumped immediately into this project she's going to tell you about today, that I will admit, when she started, I think I admitted to her I thought was not going to work out. And so I said that we're going to think of backup plans, and if this doesn't look good in a couple months, then we're going to do something else. But it's turned out really beautifully. It's a lovely project, and it's currently up on bioRxiv, so you can see all of the data in detail if you want to look there. And Angelica's done a wonderful job really fleshing out everything from a really cool phenotype to a molecular mechanism, understanding how neurons are sensing and responding to demand for mitochondria to change the rate of mitochondrial biogenesis. So with that, I will let Angelica tell you about her project.
Thanks, Katie. So thank you all for coming. I'm excited to talk to you about my thesis project looking at how mitochondrial retrograde transport regulates mitochondrial biogenesis in zebrafish neurons. So a little background, our nervous system is really important for our function. It allows us to think, it allows us to move, and allows us to perceive and interact with the world around us. So it's really important that we maintain the health and function of the cells in our nervous system, our neurons. And this is a challenge because unlike most of the cells in our body, the cells in our brain and our spinal cord cannot regenerate. So the ones we have during development are the same ones we have when we're 80. So a big question in the field of neuroscience is how do we maintain these neurons in a healthy level throughout a long lifespan? And we know that's important because if we don't maintain neurons, it leads to neurodegenerative diseases like Parkinson's and Alzheimer's. And these diseases are becoming more and more of an issue as lifespan increases and outside factors become more of a risk. So one of the goals in the field and a goal of our lab is to better understand how to keep our neurons healthy and functional. And one really important aspect of that is mitochondrial function. Our neurons are very energetically heavy cells. They require a lot of energy to function, particularly for neurotransmission, which allows our neurons to talk to each other. And so the neuron not only has to maintain the mitochondria in kind of the central cell body location, but also in very distal regions of the neuron. So neurons send out these long projections to different aspects of the body to help branch onto other neurons and form synapses. And at these distal regions, like the axon terminal, we need mitochondria to produce ATP for neurotransmission. They also have important roles in calcium buffering. So it's really important for the neuron to maintain these distal mitochondrial populations. The thing that makes that tricky is that some of these extensions are very, very long, such as the ones in our sciatic nerve, which can be up to 1 meter long from the cell body to the distal axon. So an open question in the field is how is the neuron able to sense these very distal populations and maintain them? So that is what we use the zebrafish to better understand. So we like the zebrafish because we really focus on microscopy in our lab and being able to study mitochondrial populations in vivo. And so here we have a picture of a 4-day-old larval zebrafish. Its nervous system is expressed with a green fluorescent marker. You can see the fish brain up top. You can see its spinal cord. And then we focus on the zebrafish posterior lateral line neurons. So these neurons cluster their cell bodies in a ganglion near the ear and head of the fish. And then these cell bodies will send axon projections down the length of the fish. which form synapses through these axon terminals that they form. And we like these neurons because they're very superficial, so we can easily image them. And they also, since they have these long extensions that in some cases extend all the way down the tail, we're able to kind of get at this idea of proximal versus distal in the neuron. And so we can also express, for example, a mitochondrial marker, which is shown here. So here we have the full ganglion with all the cell bodies. And then in one of these cell bodies, we're expressing a mitochondrial marker. And we can see that in the cell body as well in the axon terminal. So previous work in the lab has used these neurons through a genetic screen and identified a mutation in the gene ACTR10. And this mutation leads to the formation of these distinctive axon terminal swellings. So when we look to a wild type axon terminal, you can see it's a lot more filamentous. It has this distinct basket-like structure. Whereas in the mutants, we get these swellings that form. And previous work in the lab showed that these swellings are just full of mitochondrial accumulations. So other components of the neuron seem pretty normal, but just mitochondria seem to want to accumulate out in these axon terminals. And we can quantify that accumulation through the measure of mitochondrial density, which is just a measure of mitochondrial area divided by cytosolic area. We see that significantly increased in these axon terminals. So what is ACTR10? It is a component of the dynein-dynactin motor protein complex, and it's required for retrograde cargo transport in a cell. So in a neuron in the axon, cargo is transported in kind of the same direction by different motors. So anterograde transport is going to be from the cell body to the axon terminal, whereas retrograde transport is from the axon terminal back to the cell body. So ACTR10 is part of the complex that will move cargo retrogradely. So we thought if normally ACTR10 is important for the cargo transport of mitochondria, if we're losing that transport, it'll cause mitochondria to accumulate in the axon terminals. So to test if that was true, we used mitochondrial transport imaging. So we can measure, image just a short stretch of the axon here in the body of the fish. And then we can label our mitochondria, which are labeled here in white, and we can see the direction they're moving. So cell bodies are on the left, so anterograde transport will be to the right, retrograde to the left. And then we can compare that movement in a wild type neuron. So we can see some mitochondria moving to the right, and then in a couple of seconds, you'll see some come here from the right to the left. Whereas in ACTR10 mutants, we do still see that anterograde transport to the right, but we very rarely see the retrograde transport, and that's quantified here. We see an overall reduction in the percent and number of mitochondria moving retrogradely. So this supports the theory that what's happening in our ACTR10 mutants is that mitochondria are failing to move retrogradely, leading to these axon terminal swellings and accumulations. But the other really interesting thing about these mutants, and the thing that kind of sparked my project, was noting that in the cell body, the mitochondrial population seemed to be reduced. So that's quantified here through a measure of mitochondrial density. So here, instead of looking at the whole ganglion, we're just looking at one single cell. And we see that in a wild type cell body, the cell body is outlined in white. Here, it's in green up top, and then the mitochondria are in magenta and white here. And you can see that they fill the majority of the cytosolic space in the wild type cell body. There's just a space for the nucleus. Whereas in the mutants, we lose a lot of that mitochondrial population. So we were interested in understanding what is it about these mutants that's preventing them from kind of maintaining wild type levels of mitochondria in their cell body. So for that, we had to think about how mitochondria are normally regulated in neurons. So mitochondria are produced through the process of biogenesis. You can't make a mitochondrion de novo. They have to be produced through the growth and replication of existing mitochondria. And this requires protein gene expression, and the majority of mitochondrial proteins are actually encoded by the nucleus. And so it's thought that the majority of biogenesis in the neuron happens in the neuronal cell body. And then these mitochondria that are produced through cell body biogenesis get transported to more distal regions of the neuron, like the axon terminal. But the question is, since we know that these distances between the cell body and the axon terminal can be very far apart, how is the mitochondria able to actually sense what's happening in these more distal regions? And how does the neuron then react to either needing more or less biogenesis in these distal regions? So based on the phenotypes we were seeing in our ACTR10 mutants, we hypothesized that it was the retrograde transport of mitochondria back to the cell body that was helping to regulate the amount of cell body biogenesis. So to test that, we had to dig in a little deeper to the different aspects of mitochondrial biogenesis. So when you're replicating a mitochondria and producing more, you first have to have nuclear gene expression of mitochondrial proteins. So this includes the expression of things like the OXPHOS subunits, so the components of the electron transport chain that allow for ATP production. And then mitochondria also have their own DNA, and their DNA is actually regulated by nuclear encoded machinery. So you have to produce all the components to regulate both the mitochondrial DNA transcription and the replication of that DNA to produce new organelles. So one component of biogenesis we could look at is the mitochondrial DNA replication. So anytime you're producing new organelles, you want to maintain that genome and replicate the DNA. So for this, I used a marker of mitochondrial DNA replication, SSBP. This is called the mitochondrial single-strand binding protein, so it'll bind to the DNA strands as they separate to undergo replication. And other groups have shown that this marker nicely localizes to nucleoids that are actively undergoing replication. So this is what this SSBP protein looks like in our cell bodies. So on the top, we have a wild type neuron, and on the left, you can see the mitochondria in the cell body. And you can see these puncta within it, which is these SSBP protein puncta, and these are going to be the sites of active mitochondrial DNA replication. And you can see between wild type and mutants, we have a significant reduction in the number of mitochondria that are undergoing mitochondrial DNA replication, suggesting we do have a loss of this key component of biogenesis. I wanted to confirm that the change we were seeing wasn't due to general loss of nucleoids. I wanted to see if it was actually the replicating ones that we were losing. So I used a more general marker of mitochondrial DNA, which is TFAM. TFAM wraps around mitochondrial DNA, kind of like a histone in the nucleus. And so it's a nice marker of all DNA nucleoids. And when I compared nucleoids in a wild type versus ACTR10 mutant, I found that there was not a loss of these TFAM puncta. There's actually a bit of an increase in the number of puncta relative to mitochondria in ACTR10 mutants. So we're not losing all DNA nucleoids, we're just losing the actively replicating ones. So it's one aspect of biogenesis that seems to be disrupted. I wanted to look a little upstream and look at what was happening with the regulation of nuclear gene expression. So the protein that is thought to regulate mitochondria gene expression and is thought of as kind of the master regulator of biogenesis is this PGC1A protein. It can be activated by upstream signals in the cell that might tell it that it needs more mitochondria. So things that will sense low energy levels like the AMP to ATP ratio, the NAD plus to NADH ratio, as well as things like calcium levels. These can all activate PGC1 alpha, which then binds to transcription factors and leads to the expression of nuclear genes. And this includes TFAM, which is the protein I just mentioned. And so as a readout of how much transcriptional regulation we were having for biogenesis, I focused on the gene expression of these two key markers, PGC1A and TFAM. And to quantify the expression of these markers, I turned to a method of quantitative RNA fluorescent in situ hybridization called HCR. And so the way this works is you have a fluorescent probe that will recognize your mRNA of interest, in this case, PGC1A and TFAM mRNA. And then you can quantify the amount of mRNA present in your tissue of interest based on the fluorescent intensity of these probes. And so here I have a whole ganglion from the lateral line neurons and a wild type and ACTR10 mutant ganglion. And so I could compare the expression of these two markers between wild type and mutants. And I found that there was a significant reduction of both PGC1A and TFAM expressed in our mutants, again, suggesting that this key regulator of biogenesis was disrupted. So I'll pause for a second here and talk about an RCR component of my research, which is data acquisition and analysis. So when I'm taking images like this on the confocal, trying to quantify fluorescence intensity of something, a few things I keep in mind for data acquisition is that I need to keep microscope laser power and detector gain consistent, so I'm quantifying true differences in signal. And then because I'm working in a whole fish and I have variability in both kind of mounting of the fish and in tissue permeability. I always normalize the fluorescence intensity to background expression. And then a few other components of analysis is using biological replicates. So when I show a single data point, that's indicative of a neuron from a single fish. And so I image multiple fish to get all my data points. And then I also do experimental replicates. So I replicate the same experiment over multiple days to avoid day-to-day variability. So just a little summary of what I've seen so far. In our ACTR10 mutants, where we see loss of mitochondrial retrograde transport that leads to reduced markers of mitochondrial biogenesis, including nuclear regulators of mitochondrial gene expression, mitochondrial DNA replication, and ultimately we're seeing loss of cell body mitochondrial density. But I know that there are a lot of mitochondrial proteins, and I know PGC1A regulates a lot of things. So I wanted to be able to look more broadly at how the genome was affected, or the transcriptome was affected in ACTR10 mutants. So for that, I wanted to do an RNA sequencing experiment, which was a bit of a challenge in our fish because we're trying to look at this very specific population of neurons, and we want to be able to isolate them. So with the work of a lab tech in our lab, we developed a line of zebrafish that expressed GFP specifically in our lateral line neurons. So I could then perform embryo dissociation and use fluorescent activated cell sorting to sort out those GFP positive neurons. There are only about 200 of these neurons per fish, so I had to do this many times over many months to get enough tissue to pool and do bulk RNA-seq, but I was able to get enough to do 3 replicates of each phenotype, and then do RNA-seq and differential expression analysis to identify genes that were differentially expressed in our ACTR10 mutants. And so I anticipate with the loss of mitochondrial biogenesis that in our mutants, a lot of our downregulated genes are going to be mitochondrial-related genes. And so to test that, I used KEGG enrichment analysis of my significantly downregulated genes to see what pathways were affected. And the most severely affected pathway was a mitochondrial-related pathway, oxidative phosphorylation, which gave me confidence that what we were seeing was an effect on biogenesis. Similarly, I did GSCA analysis to look at enrichment and found that there was the top de-enriched pathway was mitochondrial protein complexes, and we can see that across the board, we're seeing down regulation of mitochondrial genes. So this is providing strong evidence that in our ACTR10 mutants, when we're losing mitochondrial retrograde transport, we're losing mitochondrial gene expression, and this is resulting in the loss of biogenesis that we're seeing. However, I wanted to be able to confirm that the changes I was seeing were in fact due to the changes to retrograde transport that we were seeing and not some sort of off-target effect with the mutation I was using. So I wanted to try and replicate the biogenesis phenotypes without using the ACTR10 mutants. So the way ACTR10 is working to disrupt retrograde transport and the movement of mitochondria back to the cell body is just to take out this kind of truck that moves mitochondria. The other way you could disrupt it is just to kind of force mitochondria to move in the anterograde direction, reducing the retrograde pool. And so for this, I used an artificial tether that was developed by a lab at Yale in C. elegans. And so the way this works is you take a mitochondrial outer membrane localization sequence and you tether that to a kinesin motor. So a kinesin motor is what moves cargos in the anterograde direction. So by tethering them together, you're essentially forcing the mitochondria to move anterogradely. And then I can visualize it through this GFP that I have connected to the tether. And so when I express this tether, it works the way I expect. So when I look in the axons, I see this mitotruck in green, localizing to my mitochondria in magenta. And I see it forming these large accumulations similar to what I was seeing in my ACTR10 mutants. And when I look at the cell body, I see a similar reduction in mitochondrial density that I was seeing in my mutants. So unlike wild type, where the mitochondria are kind of filling the whole space, we're seeing a significant reduction in the mitochondrial density in these cell bodies that are expressing this mitotruck tether. So I then wanted to look at different aspects of mitochondrial biogenesis to see if this disruption to transport was disrupting biogenesis. So I went back to my HCR RNA fish to look at nuclear gene expression of TFAM mRNA. So previously when I was doing this, I was looking at a whole ganglion. Here I'm just looking at individual cell bodies that are expressing either just flat GFP or the mitotruck. And we do see that in our mitochondrial neurons, we have a significant reduction in TFAM gene expression. I then use SSBP as a marker of mitochondrial DNA replication to see how much our mitochondria were undergoing mitochondrial DNA replication, and again, found a significant reduction in SSBP puncta, suggesting that two core aspects of mitochondrial biogenesis were disrupted without using this ACTR10 mutant, which gave me more confidence that the effects I was seeing were an effect of mitochondrial retrograde transport. But there's kind of two ways of thinking about how mitochondrial transport could be influencing mitochondrial biogenesis. So the way we'd kind of been thinking about it is that you have these mitochondria out in the axon terminal, they're coming back to the cell body, and that's serving as kind of an active signal to the cell to trigger mitochondrial biogenesis. But there's another way you could think about it. Potentially, this is just an off-target effect of disrupting retrograde transport. Maybe that's affecting some aspect of mitochondrial dynamics, and this is leading to kind of indirect effects on biogenesis. So when you look in a wild type cell body, you can see that the mitochondria form these really cool complex networks. So it's very different from kind of how we draw mitochondria in examples. They're not necessarily independent organelles. They form complex networks, and they're constantly undergoing these fission-fusion dynamics to separate from each other and then fuse back together to kind of interact as a population. And so something that's always kind of stood out in our ACTR10 mutants is that compared to wild type, the mitochondria do look potentially larger and rounder, which could be indicative of altered fission-fusion dynamics. But it's a little hard to tell at the confocal level and light microscopy level. So we decided to look at our cell bodies using electron microscopy to get a better idea of mitochondrial structure and morphology. And so when we look in a wild type cell body, we can see that the mitochondria are kind of spread all throughout the cytosolic space. And you can see these nice cristae within the mitochondria. And you can see even in, so in this case, we're taking a section through the ganglion and looking at individual cell bodies within the ganglion. So in the case of this one, where we have a section with very limited cytosolic space, we still see mitochondria. And I actually saw no cell bodies where I couldn't see a mitochondrion in that space, suggesting they're pretty evenly distributed in a wild type cell body. Whereas in ACTR10 mutants, the distribution was a lot more variable. So you'd have some cell bodies like this, where maybe some of the mitochondria were a little longer and more interconnected, but for the most part, they looked pretty wild type. They were spread out. They were in various shapes and sizes. Whereas others, I actually couldn't see any mitochondria present, and this was true in quite a few cell bodies that I was looking at. And then in some cases, I saw what I was occasionally seeing on the light microscopy level, where we have these significantly enlarged mitochondria. And despite being enlarged, they do actually have pretty normal, healthy-looking cristae. But we do see on average that mitochondrial area is enlarged in these ACTR10 mutants. So that made me want to test this hypothesis that retrograde transport could be disrupting something about mitochondrial dynamics and that this could have an effect on biogenesis. So I asked the question, could just altering fission-fusion dynamics alone lead to mitochondrial biogenesis defects? So the way I went about this was to focus on this fission factor, DRP1. So DRP1 is critical for mitochondrial fission. It's a protein that wraps around the outside of the mitochondrion and constricts to result in fission. And if you disrupt DRP1 function, you disrupt fission, leading to enlarged organelles like I've seen in ACTR10 mutants. And so we disrupted DRP1 using a dominant negative form of the protein. And you can see, compared to wild type, we do have these enlarged organelles, and we see a significant reduction in mitochondrial density. But interestingly, when we do this in ACTR10 mutants as well, we're actually further disrupting kind of the mitochondrial shape and decreasing density even further, suggesting that these are working in independent pathways. But we could still have effects on fission that are disrupting biogenesis. So I wanted to see the effects on some of these markers of biogenesis. So I first looked at TFAM gene expression in wild type control, ACTR10 control, and then in DRP1 disruption. And so we can see, as I've seen previously, that when you compare wild type to ACTR10, we have a reduction in TFAM expression, But when you add in this DRP1 disruption, it doesn't affect TFAM expression. The wild type looks like wild type, ACTR10 looks like ACTR10. Similarly, if we disrupt DRP1 and look at mitochondrial DNA replication with SSBP staining, we can see that even though we're kind of altering the morphology of these mitochondria and the nucleoids are getting a little more condensed, the number of DNA nucleoids that are actively replicating does not change between control and DRP1 disrupted mitochondria, suggesting that just disrupting mitochondrial dynamics alone is not sufficient to affect biogenesis and better supporting our original hypothesis that the return of mitochondria to the cell body is a more active signal to regulate biogenesis. So that still leaves the question of what are these mitochondria doing when they come back? So to start to get at that question, I focused on one aspect of biogenesis, which is the regulation of nuclear gene expression. So because I have my RNA-seq data, I know that a lot of mitochondrial genes are downregulated in ACTR10 mutants. So I hypothesized that there could be a common transcription factor that was required to regulate the expression of these genes that was being disrupted in our ACTR10 mutants, leading to reduced expression of these genes. And so I performed transcription factor enrichment analysis of my significantly downregulated genes and identified transcription factors that are known to bind in the promoters of these mitochondrial genes. And the two that popped out was this ESRRG and ESRRA, which are estrogen-related receptors. And not only were they significant in my data set, but they've also been previously linked to mitochondrial biogenesis. So there are a few different transcription factors that are known to induce expression of mitochondrial genes. And ESRRA and G are one of these. They're known to bind to PGC1A to help regulate their activity. And they induce the expression of some of the genes that I know are affected in my data set, like these oxphos proteins and TFAM. And I felt a little more confident that it could be these specifically because some of these other known regulators of mitochondrial genes like NRF1 and PPAR didn't come up in my transcription factor enrichment. So I felt a little more confident that these estrogen-related receptors might be a specific thing that was being regulated, and that mitochondrial retrograde transport could somehow be feeding into their activity either through regulating the upstream PGC1A or by activating them directly. So my hypothesis is that in ACTR10 mutants, we have impaired activity of estrogen-related receptors and that this is causing reduced mitochondrial biogenesis. So we'd hypothesize if we can just go in and rescue ERR activity, it'll rescue mitochondrial biogenesis. And so to do that, we first wanted a way to increase ERR activity. So for that, we overexpressed the most kind of well-known estrogen-related receptor, which is ESRA. And we found that in a wild type background, when you look at TFAM mRNA, we can significantly increase that gene expression with ESRA overexpression. And we can also increase overall mitochondrial density in the axon terminal, suggesting we are inducing mitochondrial biogenesis. So what happens if we overexpress ESRA in an ACTR10 background? We know previously that TFAM is normally reduced in ACTR10 mutants, but with ESRA overexpression, we're actually able to rescue TFAM expression back up to wild type levels. Similarly, we can actually bump up the mitochondrial density in the axon terminals to higher levels than we're even seeing in our mutants, and to the same level that we see in a wild type background, suggesting that we can, in fact, rescue these biogenesis deficits with ERR activity and suggesting it is downstream of this mitochondrial transport disruption. So then we wanted to see if we could go even further and identify kind of the upstream signals that were helping to regulate ERRs and regulate biogenesis. So we decided to study two kind of well-established pathways that regulate biogenesis and regulate these transcriptional mechanisms. So both of these pathways are regulated by kind of upstream energy levels. So they're kind of the energy sensors in the cell. We have SIRT1, which is regulated by the NAD+ to NADH ratio, and then AMP kinase, which is regulated by the AMP to ATP ratio. So first, I wanted to see if AMP kinase could be upstream. It's known to activate PGC1A to induce ERR activity, so it could explain if AMP kinase is not being activated, why we are losing biogenesis in ACTR10 mutants. So I decided to, similar to my ASRRA overexpression rescue, try and rescue biogenesis in ACTR10 mutants by artificially inducing AMP kinase activity with the drug ACAR. And so when I use this drug treatment in wild type versus ACTR10 mutants, I don't actually see any change in cell body mitochondrial density. So we don't seem to be able to induce mitochondrial biogenesis just by activating AMP kinase. Excuse me. So it doesn't seem to be AMP kinase that's key. Maybe it's SIRT1 instead. So we similarly induced SIRT1 activity using the drug resveratrol. And in this case, I was actually able to rescue ACTR10 mitochondrial density. So if you compare an ACTR10 normally to the resveratrol treated, we see at least a partial rescue of mitochondrial density closer to wild type levels of mitochondria. So it suggested to us that SIRT1 signaling could be a critical link between mitochondrial retrograde transport and activating these downstream transcriptional pathways through PGC1A and ERR. So normally SIRT1 is regulated by these NAD+ to NADH ratios. So we hypothesized that there could be something about the mitochondrial return to the cell body that was altering these NAD+ to NADH ratios. Maybe the mitochondria that are normally coming back to the cell body have a higher level, which elevates the cell body levels of NAD+, activating SIRT1 and inducing these downstream biogenesis factors. So to test that theory, we used a genetically encoded biosensor for NAD+, which we localized to the mitochondrial matrix, which allowed us to specifically look at NAD+ levels within the mitochondria in different regions of the neuron. And with this, we were able to compare mitochondrial NAD+ levels in the cell body versus the axon terminal for wild type and ACTR10. And so the way this works is that higher NAD+ levels are shown in kind of a yellowish white color, whereas low levels are shown in this bluish purple color. And so you can see when you look at the cell bodies between wild type and ACTR10 mutants, we have lower NAD+ levels in the cell bodies. Whereas when you look in the axon terminals in the mitochondria, so the mitochondria are outlined in this kind of dotted line within the overall terminal. And you can see that the ACTR10s have higher levels of NAD+ within their axon terminal mitochondria. And those are both quantified here. So this suggests that ACTR10, we're losing the NAD+ high population. It's kind of getting sequestered out in the axon terminals and not coming back to the cell body. And so this is kind of my proposed model of how the neuron regulates biogenesis through retrograde transport. So we have mitochondria with higher NAD+ to NADH ratios that are out in the axon terminal. Those mitochondria get brought back to the cell body, where they then locally elevate NAD+ to NADH ratios, activating SIRT1 activity to induce mitochondrial gene expression and ultimately induce mitochondrial biogenesis. So this gives us a better understanding of how the neuron is able to sense mitochondria in very distal regions and kind of respond to differing needs of the axon terminal to kind of replenish that population through cell body biogenesis. And this is important because it gives us a better understanding of how the neuron can maintain its mitochondrial population and potentially maintain health and function. So with that, I would like to acknowledge the Jurup Lab and thank them for all their help, also recognizing my committee, my funding sources, and the genetics program. And as Katie mentioned, my work is now up on bioRxiv, so if you're interested in learning more, you can scan that QR code. So now open to questions. Okay.
I was just wondering, normally, what happens to the mitochondria that are retrograde transported? Do they undergo mitophagy, or do they just get incorporated into the network and somehow whatever is not working well gets turned over? How is that managed in this?
Yeah, so kind of like the canonical thought in the field was that mitochondria were only coming back to get degraded, but the work by our lab and others suggests that the mitochondria actually get maintained that are moved retrogradely, so we don't quite know what they're doing. They are coming back and they seem to persist in the population over very long periods of time. And so we do think they're kind of coming back and fusing back in with that network. Maybe it's a way to kind of help maintain health, like they come back and kind of replenish mitochondrial proteins through that fission and fusion with the cell body network and through the biogenesis that gets activated there, and then they can go get shipped back out to maintain the distal population.
I'm sorry, would you try to repeat the question?
Oh, yeah. So the question was just like, what is the purpose of mitochondrial retrograde transport? What do they do?
Yeah, I was wondering, do you see elevated levels of that? Because there must be some sort of degradation, is it proteins or anything that's not functioning now? Do you see evidence there?
Yeah, so we know that some of the organelles that come back are getting degraded. So they generally get loaded into autophagosomes to get degraded out on the axon terminal. And it's thought that the retrograde transport is in part working to kind of bring them back to the cell body to get fully degraded. And then, sorry, can you repeat your question?
But in terms of recycling proteins that might be damaged.
Yeah, so that's what.
Do you have any insight into how those specific molecules would get exchanged in the mitochondria?
Yeah. Yeah, so one thing we do see in our ACTR10 mutants is the mitochondria that are stuck out in the axon terminal do have lower measures of health. So they have like higher ROS production and lower ATP production, which suggests they're more unhealthy when they're stuck out there. So there does seem to be something about the retrograde transport that's important for kind of maintaining the mitochondria, but we don't exactly know specifics of how it's doing that.
And maybe this is a related question. I was gonna ask how well understood it is. How are the mitochondria and the terminus identified for retrograde transport? How are they selected?
Yeah.
Who decides who gets on the truck, I guess?
So the question is, how do mitochondria know to move retrogradely? And that's something we don't really know, and is something we're really interested in this lab, is this idea of like cargo selection, because we know some of the components of mitochondrial transport. We know the motor that they get transported on, and we know the some of the adapter proteins that connect mitochondria to the motor to move it retrogradely. But what we don't know is what kind of determines that selection, and something our lab is interested in is a role for ACTR10 in kind of regulating which mitochondria get loaded on. We think it might be serving as kind of a help load the mitochondria onto this motor to move retrogradely rather than being like the sole thing that's like moving them. Yeah.
I have two questions. First, why is resveratrol only partially rescuing your ACTR10 phenotype? Do you think there's some other factor in play as well downstream of ACTR10? My second question is, do you have any speculations as to why the mitochondria at the axonal terminal are like have higher or lower NAD plus over NADH ratio versus the one in the cell body?
Yeah, so the first question was why we're only seeing partial rescue with resveratrol drug treatment. And so with that, we think there could be a few things at play. We're only treating them for like a small period of time, so maybe it's not enough to get them back to normal levels, or it is likely that there probably are other pathways that are interconnected here that we're not fully rescuing with resveratrol treatment. That's something that we're kind of interested in the lab, is other mechanisms that might be important for regulating biogenesis. And then the second question is why the mitochondria that are out in the axon terminal and moving retrogradely might have higher NAD+ levels. I don't have a great answer to that question. We know that there are kind of like different populations of mitochondria that might have different roles. And there's been some evidence by other groups that some might be more kind of important for biogenesis factors, for example. But yeah, I don't quite know what it is about these mitochondria that are coming back that would why they would have higher NAD+ levels.
Do you see like something similar in other cell types apart from neurons, like where like two different compartments of cells have mitochondria with varying NAD+ levels, or is it only specific with this?
I've only looked in the specific cell type. A lot of other non-neuronal cells are much like smaller and rounder, so there's less need for this kind of like active transport to regulate. If you just have NAD+ levels in that cell, it can directly activate cell bodies. But I would be interested to look in other neurons in particular and different subtypes of neurons to see if this is a kind of conserved phenotype. Yeah.
What is the fate of the neurons in the ACTR10 mutants? If it's accumulation of mitochondria at the end, what happens to them?
Yeah, so the neurons do actually seem to be functional. So we've looked before at their ability to like have like synaptic activity, and that seems to be preserved in these mutants. The neurons do survive. The fish have other defects, so the fish end up dying around 8 to 10 days, I think. We can't look at the neurons past that point to see what would happen, but up until that point, the neurons we don't see like evidence of neurodegeneration, for example, like you see with other retrograde transport defects.
Is there any evidence of recycling of mitochondria at the axon terminal because of this accumulation?
Yeah, so there is, we have looked at mitophagy, which is how mitochondria get degraded in these neurons previously, and they do seem to be able to get degraded. So there does seem to be like some baseline levels of degradation happening, but it seems to be insufficient to keep up with how many are out there, and like potentially it's only being induced by the really unhealthy ones, whereas the ones where we're seeing like partial like signs of failed health, they don't seem to be getting degraded.
Right? No more questions? Let's thank Angelica again.
Thanks, guys.